Hello everyone, my name is Yangyang Hu from UC Riverside. Today I will present EnHiC, learning fine resolution high-C contact maps using a generative adversarial framework. In the presentation, I will first introduce high-C data and the fine resolution problem. To solve this problem, we proposed a neural network named EnHiC. We compared our model with previously proposed GAN-based neural networks and evaluated them in the three cell types. The predictions from our model also performed well in TAT detection and identification of significant interactions. Recent developments of the high-throughput chromosome conformation capture techniques, HiC, have enabled us to detect genome-wide chromatin interactions and investigate the organizational principles of the three-dimensional genome. Studies of HiC data have relieved the multi-scale organization of the 3D genome. AB compartments, topologically associated domains, we call TADs, can be identified at a relatively low resolution from 50 kilobase resolution to 1 megabases resolution. Fine-scale chromatin loops require high-resolution HiC data. Here is an example of HiC matrix. HiC matrix is an example of DNA-DNA contacts from thousands to millions of cells. It is a symmetric non-negative square matrix. The values indicate the number of contacts between all pairs of loci. The pixels in the heat map from blue to red represent the values from low to high. High-resolution HiC data requires costly deep sequencing. Therefore, it has only been achieved for a limited number of cell lines. Without sufficient sequencing depth, the observed HiC matrix is often sparse and noisy, which imposes great computational challenges on the identification of chromatin loops. Therefore, computational approaches to enhance the resolution of HiC matrix would greatly facilitate the investigation of the 3D genome at a finer scale and are therefore in great demand. Our goal is to predict high-resolution sufficiently sequenced HiC matrix from insufficient sequencing data. Here, high resolution indicates more chromatin interaction details, rather than a higher dimension of the HiC matrix. Given a HiC input dataset, it can be processed into a matrix of any arbitrary bin size. Therefore, a high-dimensional HiC matrix is not always of high resolution. In this work, we refer to the dimension of HiC matrix as its scale. A lower scale HiC matrix has a smaller number of rows and columns. Sequencing depth describes the number of contact pairs observed in the experiment. Here is an example. The top left one is the high-resolution sufficiently sequenced HiC matrix. Three HiC matrices from left to right are from the same input. The bin size is increasing and the resolution decreasing. Meantime, the size of matrices shrink too. The left one is 400 by 400, and the right one is 50 by 50 at 80 kilobases resolution. The insufficiently sequenced HiC matrices are sparser and noisy. The value in the matrix is much smaller than high-resolution HiC matrices. We can simulate the insufficiently sequenced HiC matrix by downsampling rates. Here is the relation between resolution, dimension, and sequencing depth. The top left one is a high-resolution sufficiently sequenced HiC matrix containing more details. The bottom one is a high-dimensional HiC matrix at low resolution, and both of them can be resized into a larger bin size. The top left HiC matrix is the high-resolution HiC matrix, and the rest three are all at low resolution. The left one is the input of model. Several methods have been developed to enhance the resolution of HiC matrix. HiC Plus and HiC-NN are two convolutional neural networks to enhance the resolution by minimizing the L2 loss function. However, L2 loss function is sensitive to the outliers and suffers from the sparse HiC matrix, resulting in a blurred output. The previously proposed models, HiC-GAN, Deep HiC, and HiC-SR, are demonstrated the power of GAN framework in predicting high-resolution HiC matrices. These models treat the HiC matrix as a one-channel image, and their GAN networks are built on image super-resolution models. As a result, their predictions often contain artifacts, and therefore do not accurately represent the underlying chromatin interaction features. To tackle this problem, we developed a new GAN-based model, EnHiC, to enhance the resolution of HiC data. In computer vision, convolutional neural network discovers the local features in the image. Here, we treat HiC matrix as a summation of contact maps rather than an image, and utilize the properties of HiC matrix, non-negative and symmetric. It not only discovers the local features, but also extracts the features from the third dimension, dimension of cells. Specifically, in our GAN framework, the generator extracts rank 1 features from multiple scales of low-resolution matrices and predicts the high-resolution matrix via subpixel convolutional neural network layers. The discriminator extracts the corresponding rank 1 features in multiple scales to determine whether the input is derived from generator or the true data. Here is the idea of our model. The input is sparse, so we first resize the input into multiple scales by using space to depth. Space to depth will shrink the matrix and extend the number of channels. Then we extract rank 1 features. Based on the rank 1 features, the model uses convolutional neural network layers, CNN, to detect local features, and upsampling them back to high-resolution matrix as output. The rank 1 matrix maintains the symmetric property, and activation function ReLU guarantees non-negative property. Next, we introduce the details of extracting rank 1 features. In this case, rank 1 matrix is the outer product of one vector and its transpose. This layer outputs a set of rank 1 matrices. A weighted summation is to approximate the low-resolution HiC matrix. Here is the implementation. We customize the kernel size of 2D convolutional neural network layer. The input is X, and the model learns parameters H to generate the set of vectors V. The outer production of one vector is one rank 1 matrix. The non-negative matrix decomposition finds rank 1 matrices for one matrix, but here we find rank 1 matrices for a set of matrices, and the difference is defined by loss function. Then we pass the rank 1 features to subpixel layer, which is closely related to the image super-resolution. The channels will be shuffled into one. For example, there are 3x3, 9 channels. Colors means different channels: red, yellow, green, cyan, blue, purple. The pixels will be paved in order: red, yellow, green, cyan, blue, and purple. And here is another repeat. Similarly, for HiC data, 4x4, 16 channels at 40 kilobits resolution will generate one matrix at 10 kilobits resolution. Here is the framework of generative adversarial network. The generator has two parts: extracting rank 1 features in part 1 and the upsampling in part 2. The model updates two parts separately. For the part 1 in orange, the model applies structure similarity loss function and L2 loss function to approximate low-resolution HiC matrix. The part 2 uses L2 loss function and adversarial loss function for predictions. Based on adversarial gaming, the adversarial losses are negative log likelihood loss function in generator and discriminator. In the evaluation, we compared EnHiC with Deep HiC and HiC-SR which both are GAN-based models. We trained the three models on cell line GM12878 from Cooler database at 10 kilobases resolution. The low-resolution HiC data is at 40 kilobases resolution, generated by downsampling. Chromosomes from 1 to 16 are in the training dataset, 17 and 18 are in the validation dataset, and the rest 5 chromosomes are in the test dataset. Table summarized HiC-Rep genome disco evaluation results. As shown in the table, the HiC-Rep scores were greater than 0.94 for all three methods, indicating the high-resolution predictions are very similar to the ground truth. EnHiC model achieved the highest scores in both metrics for all 5 test chromosomes. In addition to the evaluation on one cell line, we applied 3 trained models to 2 other less sequenced HiC datasets. The previous training dataset has highest number of chromatin contacts. The figures illustrate cross-cell type performance of 3 models. Overall, EnHiC outperformed both DeepHiC and HiC-SR. We observed that the scores were relatively lower than the one previous obtained dataset, but they were significantly higher than the baseline, the low-resolution HiC. In addition, the performance of all 3 models were slightly better in IMR90 than K562. This is likely due to the relative higher sequencing depth. Taken together, these results indicate that EnHiC can effectively recover high-resolution matrices from insufficiently sequenced HiC data across cell types. Although being trained by 16 times downsampled data, EnHiC is flexible and can be applied to low-resolution data with much less sequencing depth. We used the same trained models and then evaluated the model performance at 6 different downsampled ratios. Notably, EnHiC robustly and stably recovered high-resolution HiC matrices from low-resolution input data with large downsampled ratios. Next, we look at whether the predictions promote TAD detection. Topologically associated domains, TADs, are functional units of chromatin, while chromatin interactions are observed more frequently within TADs than outside. TAD boundaries are largely conserved across cell types and are enriched with CTCF and other chromatin-binding proteins. Here, we use the HiC find TADs method in the HiC Explorer to detect TAD boundaries. We calculated J-card score to assess the consistency between TADs detected from predictions and the detections from true high-resolution data. The left figure shows the J-card scores, and the right figure is the number of TADs detected by each model. Overall, EnHiC got highest J-card scores for 5 out of 7 chromosomes, and outperformed both DeepHiC and HiC-SR. Next, we investigated whether predictions could facilitate the identification of fine-scale chromatin loops. We applied FitHiC to identify significant interactions, and the J-card score was used to assess consistency. EnHiC outperformed other predictions. Based on the t-test, the J-card score in EnHiC is significantly greater than others. Here is an example. The left one shows the TAD detection. The top 2 figures are high-resolution and low-resolution HiC data. The next 3 are the predictions from EnHiC, DeepHiC, and HiC-SR. The right one shows the identification of significant interactions. The predictions of EnHiC were accurate. The predictions of DeepHiC and HiC-SR contained unwanted image textures, thereby resulting in inaccurate detection. To sum up, in this study, we proposed a generative adversarial framework in HiC for predicting high-resolution HiC matrices from low-resolution input data. We demonstrated the performance of the EnHiC model using EnHiC datasets on 3 human cell lines. Overall, EnHiC model evidently outperformed 2 other GAN-based models, DeepHiC and HiCSR, achieving low prediction errors and high reproducibility scores when compared with true high-resolution data. Moreover, EnHiC model is capable of recovering high-resolution HiC matrices across different cell types and from insufficiently sequenced input data. Additionally, we demonstrated that EnHiC predicted matrices facilitate more accurate detection of tags and fine-scale prompting interactions. For more details of EnHiC model, please read our full paper. That's all. Thank you for your attention.